afternoon to you all by 1 minute. Thanks very much for taking the time out to join this lightning talk. I can see from some of the names there are some frequent flyers amongst you who are obviously looking for platinum cards and attending all of the lightning talks, and a few new attendees as well whose names I don't recall from previous sessions. So all of you are very, very welcome indeed. The idea of the lightning talk is to have some really exciting speakers who span across different organizations in their responsibilities and roles, to sort of give you a really kind of punchy update or punchy angle on a particular topic. And today I'm delighted to say we've got Steve Clifford and Ruth Plummer. Steve is the director of the Newcastle University Centre of Research Excellence in Cancer, and Ruth Plummer is a clinical academic who is also director of the Sir Bobby Robson Cancer Research Centre, which is funded through CRUK, and perhaps more of that anon, but that's been quite a notable achievement recently to have the funding for that renewed, as you may have seen. So without any further ado, Ruth and Steve are going to do a double act, starting with Steve. They're going to be done in 15 minutes or so, and then there'll be time for questions. In the time-honored way, you can either do that by raising your hand or by all means pop your question into the chat. Over to you, Steve.
That's great. Thanks very much indeed, David. And above all, thanks for the invitation to come and be able to talk to you all about the cancer research that we're doing here at Newcastle University. So first of all, why are we here? This is the need and the numbers. And I think it goes without saying, over 300,000 new diagnoses of cancer every year. One in two of us can expect to be diagnosed with a cancer right now in our lifetimes, and 1 in 4 of us will ultimately die from the disease. So a major public health challenge. This year represents the 40th anniversary of cancer research in Newcastle. It's grown from small, enthusiastic beginnings with our first cancer research unit in the medical school. And you can see some of the highlights here taking us through the development of the Northern Institute for Cancer Research, our designation as a Cancer Research UK Centre, state-of-the-art facilities like the Sir Bobby Robson Centre and the Northern Centre for Cancer Care, Great North Children's Hospital, our Wolfson Paediatric Oncology Research Unit. This all now comes together into a number which is more like 400 or 500 researchers and 100 PIs across the university and trust delivering this translational vision.
Steve, sorry to interrupt. I don't know if others, I can't see the slides. Are people seeing the slides? Not sure you're. Yeah, there we are.
Oh, I'm terribly sorry. Right. So,
sorry, I've thrown you now.
I'll show you my slide on the needs and the numbers. I've recapped all of that, and this, as I say, are the key facilities that we've developed over the last 40 years in Newcastle, bringing us to the point of having the Centre for Cancer with over 100 PIs and perhaps 400 or 500 researchers working across trust and university for patient benefit. The core ethos of everything that we do is about the patient. It's about doing translational research to give every patient the best possible chance of a cure and the best possible chance of living a long, healthy life beyond cancer. So it all comes down to personalised medicine. And the way we personalise medicine for cancer looks something like this. We have to understand the biology and patient features, which allow us to individualise therapy. And this really means 3 things. First of all, we can de-intensify therapies for patients that are likely to do well. We can give the most intensive treatments to those that need them, and we can develop all new agents and deliver them to the right patients. And through our networks of international clinical trials, the goal, as I say, is maximal survival, minimal late effects. To do this, we really should just say that we've pulled together a whole host of partners. So sitting right at the top is the Newcastle University NHS axis, and then below.
Steve, you've muted yourself inadvertently.
Well, I think
I didn't do anything then. Something happened.
Anyway, I shall pick up there, and Steve's very kindly going to keep driving. So yes. we were asked to talk when Peter talked to us about, improving treatments for patients. So next slide, please, Steve. So, and I guess with the pandemic, we all are aware of the stages of drug development. And so I wasn't going to dwell on this because we've all heard about studies, but I wanted to point out that conceptually, to treat a patient with cancer, you've got cells that have gone wrong and you actually want to kill them. It is only the infectious disease doctors that also wish to kill cells, but they don't want to kill human cells inside the human. And that means that we actually do even our safety studies in patients, not healthy volunteers. And these are patients who know they've got cancer, they know they're going to die of cancer, and they're prepared to have an experimental treatment. Next slide, please. And what we've been doing in Newcastle is you're trying to find those better treatments. And actually, the success stories are both rucaparib and erdafitinib, two drugs that have come out of the CRUK-funded Newcastle Drug Discovery Unit, led by Steve Wedge there. And in Newcastle, we've actually got those 400-500 scientists Steve was talking about doing target evaluation, structure-based drug design, and the hit to lead pharmacology, the lead optimization, and then the first in human trials is all available in-house. Next slide, please. But I was going to tell you a little bit of the story and then how it's changed. And for me, the patients and the researchers, we're a team. And actually, to begin this story, in 2003, I met Peter, and this really speaks to me because he was in his 40s, he had a teenage family, he had an inoperable tumor, that's a CT scan with the tumor in his abdomen outlined, and he'd run out of options. So he knew there was no other option. So he was prepared to have a drug where I acknowledged him. We didn't know the dose, we didn't know what the side effects might be, and we didn't know if it was going to work. Next slide, please. And Peter was actually the second patient in the world ever to get a PARP inhibitor and to get rucaparib. And this shows the timeline, which is what I want to speak to on how things have changed. Because it was in 2003, I wrote that first prescription. And we'd been developing in the lab an assay to show that did this drug actually hit PARP, its target? And those are little bar charts from tumor biopsies our patients kindly allowed us to take, showing that even at a low dose, we were 80% inhibiting the PARP in the tumor, and that got better as we increased the dose. The other thing that got better was Peter's tumor. We don't understand why, but after 12 cycles, there's a picture of it much smaller, and he actually had a resection after 20 cycles. But this is a collaborative event, and the science didn't stop. And at the same time, the team in Sheffield had liaised with Nicola Curtin and was looking at a different way of using PARP inhibitors. We were trying to overcome resistance to chemotherapy, but they, jointly with the ICR, published in Nature the fact that if a tumor had this molecular profiling and lost BRCA, you could kill the cells. It was 10 years later that Ian McNeish presented at our big oncology conference. This waterfall plot, waterfall plots are much beloved of oncologists. Basically, you measure everything, and if it gets smaller, it goes down the waterfall, and if it grows while you're on treatment, it goes above, which you don't want to see. They saw an 80% response rate in ovarian cancer with a PARP inhibitor if there was a BRCA mutation. Next slide, please. And that really, this shows that story. We were hugely supported by CRUK over that timeline it took us. Those are the sponsors of the studies. But we got there. In 2017, it was clear we had a strongly positive phase 3. And next slide, please. The delight for me at the end of this story is the university and CRUK, who own some of the intellectual property for rucaparib, held a celebration at the Great North Museum. There was a patient engagement event, and I invited Peter and his wife along to hear the story of the drug that he'd been such a part of. And then if we then go for the next slide, please. But where Steve and I wanted to say to you is this, what's exciting is at the same time, molecular profiling, precision medicine was really coming. And these are the timelines now. If you look at that ALK timeline in the middle of the bottom of the trial, the ALK fusion was discovered in lung cancer in 2007. 2 years later, patients were being treated on a trial, and 2 years after that, the drug was approved. We are really driving these timelines, and we're doing that now with what we call basket and umbrella studies. And the poster child for a basket study are the TRAC inhibitors. TRAC fusions are rare, but it's a constituent active protein. And these investigators, largely led in the States, did a tumor site agnostic, so a basket study, all of the one type of tumor, or one type of genomic alteration goes into the basket, but age agnostic. They put children babies up to 80-year-olds on this study. And you can see a waterfall plot and what we now call a swimmer plot, the length of time people stayed on. But the other big thing that's changed are the platform studies, where you have one tumor type, but you stratify by molecular profile, and then different arms of the study all running in parallel. So you go under the umbrella. And then next slide, please. My final slide is that what are we doing that's exciting at the moment? Well, my colleague, Alistair Greystoke, is national lead for Concord, which is a big radiotherapy platform study, although he and I are medical oncologists. And that is combining the DNA repair inhibitors with a DNA damaging agent, radiotherapy, under a profiled platform. And similarly, from the Bobby Robson Foundation and Christie Charity, we funded Target National, and this really is sequencing at scale. Every patient who comes through our clinic, we're now offering circulating tumor DNA, profiling for 360 tumor mutations, and then we have across all 18 experimental cancer medicine centres in the UK now a trial finder. We know we're sharing all of our trials, we match patients to trials, and CR UK is setting up the trial called Determine, which again will offer opportunities for patients with rare mutations to get on an actionable drug. And then next slide, I'm handing back to you, Steve, to say how this has changed your world as well on the biomarker side.
My world indeed. Thanks very much indeed, Ruth. Yeah, where I wanted to pick up here is this idea of how we match the treatments to patients, because developing the treatment alone isn't enough. It's really about delivering the right treatment to the right patient. I'm going to use the example of a childhood brain cancer called medulloblastoma, which we work on a lot. You can see a medulloblastoma here growing in the cerebellum at the back of the head. The key issue here is that we deliver highly intensive therapies like radiotherapy to young children whose brains are still developing. And as a consequence, many of the patients that are cured will be cured with lifelong side effects. They may struggle at school, they may struggle to hold down jobs into later life. So this is the balance that we have to strike. We have to achieve the best cure, but we have to cure in the kindest possible way. And this is a slide which you're not meant to read in detail, but it shows how on the left in medulloblastoma, we've gone from in 2000, simply using clinical indices to diagnose these tumors, right through to the present day, where we rely on probably half a dozen different molecular indices to classify and decide treatment for these patients. The key sorts of discoveries we've made, if you look at the 4 consensus subgroups, what the disease we used to just call medulloblastoma and treat all as the same, comprises 4 molecular subgroups that you only see at the molecular level. And then critically, these subgroups are highly important clinically. They behave different clinically. And in particular, the Wnt group, which is the blue group there, you can see on the curves down on the bottom right-hand side, do incredibly well. We see almost 100% survival in those patients. So the discoveries that have been made in Newcastle have formed part of the WHO consensus classification of medulloblastoma. These are used right around the world to classify and treat medulloblastomas now. And critically, we're running trials in Europe that use these indices to tailor therapy accordingly. So this is the major European trial that's running at the moment. We take patients, we do molecular diagnostics, we put them into a low risk group. These patients have reduced radiotherapy with the goal of getting them out of the other side in much better condition, but with that maintained cure. And then in red, we have patients who need intensified therapies to give them any chance of a cure. And the side benefits of this, by running such a molecularly driven trial, it allows us to collect state-of-the-art biological material for our next analyses so that we can take things forward to the future. Of course, it's not just about delivering molecular diagnostics. To do this for a tumor type that is perhaps only seen in 3 or 4 cases in every centre has required centralised diagnostics. So in Newcastle, we host the National Diagnostic System for the whole of the UK, and we advise right across Europe. Samples come from all centres to Newcastle. We do the diagnostics. We put together high-end diagnostics together with pathology reports that go back to define patients for all across the board. We don't just do this in medulloblastoma, we have equivalent systems in neuroblastoma and with pharmacological monitoring, all driven out of discoveries in Newcastle and hosted in Newcastle. Last couple of slides, just to show you some real data. I think that's always important in a talk. The next steps with medulloblastoma is that we've been able to work on an international basis. This is an international study that we drove that's shown that subtypes that we used to think were just 2 subtypes are now 8 subtypes. Again, all with their own molecular characteristics and different clinical behaviors. And these latest subtypes have been incorporated into the 2021 classification of CNS tumors, which again is really gratifying to see from a Newcastle translational perspective. Last of all, again, not one to read, I think it's just to reinforce Ruth's point of the sheer repertoire of molecular diagnostics we're able to offer our patients now as a result of Newcastle-led research. On the left-hand side, 2014, we were doing a few key markers. Right now, we do panel sequencing, we do methylation arrays, we do a whole range of tests that are all delivered in a 2 to 3 week period that are used to define treatment. And I'll hand back to Ruth to tell us about the future.
So really, this was just next slide, please, to wrap this up. What we hope we've done is we've convinced you that we really are working. This is the CRUK goal. You know, over the 40 years, 1970 to 2010, they doubled the number of people living for 10-year survival with cancer. And within the next 20 years, they're hoping to get that to 75% of patients diagnosed. Next slide, please. And this is, you know, from, you know, Steve leads us as the director of the Centre for Cancer. And these are the bullet, you know, the strap lines we have, but absolutely this is for patients. And as David alluded to, we were delighted to be recognised by CRUK as one only 7 CRUK centres in the UK for the excellence in what we do in our sort of area of bringing benefit to patients. So thank you very much.
Fantastic. Thanks to you both, and thanks for sticking to time. It's brilliant, really good. And I have certainly witnessed firsthand at lots of different levels, including a personal level, the excellence of cancer work in Newcastle as well. So if we could come out of the screen share, that was really a tour de force. Actually, you crammed such a lot in. I'm going to ask you a very easy question, Ruth and Steve, just while everybody's thinking and putting some down. So obviously you've talked quite a lot there about precision diagnostics, precision therapeutics, novel trial designs, and I just wondered, in the next decade, what do each of you think is going to be the real big game changer in terms of breakthrough in cancer treatment? Ruth, do you want to start?
I think it is going to be that the power of the precision medicine. It's not just genomics, but that's the bit we can do. And getting that at scale, we now actually, through NHS England, could get whole genome sequencing on every single patient referred for a phase 1 trial. That is just in setup. But it's because I'd really like not to give a treatment to somebody if I could guess it wouldn't work, because you can't kill cells inside a person without some side effects. And actually, as Steve's alluded to it, you know, the burden of, almost, it's wrong to say burden of survivorship, but the burden of the toxicity we cause, you want it to be lifelong, but it then becomes a burden of toxicity. So that's the vision. It's to, how do you scale back, and how do we treat the right patient with the right drug?
Thanks, Ruth. Yeah, I mean, I, as a neurologist, certainly saw some of the consequences of medulloblastoma treated cases coming through into the adult scenario. So, Steve, what about you? Would you say the same? Or?
Yeah, I'd agree with that. I mean, as Ruth says, we fail to realise sometimes we're in a unique position in the UK with our access to whole genome sequencing. Our colleagues around Europe and around the world just simply don't have this, and we now have the capacity to sequence all children and most cancer patients, you know, in this way. So that will underpin the personalization, absolutely. We have to keep the pot bubbling on new therapies, and I think what's starting to get really exciting is looking beyond small molecules towards new types of approaches like cellular therapies and immunotherapies, and above all, the potential to combine all of these therapies together. So I think ultimately that's where the lasting cures are going to come from. And last of all, it is about kindness of treatments. I mean, we have this fantastic headline figure in kids of 85% of children now being alive 5 years post-diagnosis. But, you know, a huge proportion of those will have some form of late effect that has to be managed for the rest of their life. So, you know, that's really where we want to get to the future, that they're not just getting through, but they're coming out the other end in the best possible way.
Yeah, thanks, Steve. I agree with that 100%. So any comments or questions? Yeah, Andy, thanks very much. In you come, Andy Knight.
I only caught the end of Steve's talk, so thanks very much. It's really nice to hear that. What I wanted to ask as an immunologist and as not as a cancer biology, Steve, you alluded to immunotherapy and cellular therapy at the end. So can you just remind me where we are with those types of therapy with sort of young patients? Does that actually happen at all? I don't know the sort of story. I know that's where lots of other cancers are going.
Yeah, no,
What's the future?
absolutely. So it is happening. I think Newcastle is one of 2 sites in the UK that are delivering Genentech CAR T therapies to children with leukemia. So it is happening. I think the big step is moving to the point where we can start to produce our own CAR Ts alongside delivering CAR Ts that have come from other sites. And it's not just about CAR Ts, of course. There's a huge learning experience going on there, but a lot of small molecules which target aspects of immune and immunoregulatory pathways as well. I mean, Ruth has particular programs ongoing in these that she may want to comment on.
Well, I was going to say, Andy, I mean, one of the problems with immunotherapy and the checkpoint inhibitors is that the childhood tumors, it's been disappointing, and that may be because they tend to be a catastrophe with often one change, or not as, if you look at the adult tumors, far more small changes, therefore tumor neoantigens. So we are getting better response rates in the adult space. But yeah, no, we're participating in the Achilles study, which is a spin out from Imperial UCL, a spin out company, where these are neoantigen T cells. So they're the patient's own T cells. We send 140 mils of blood down to London whenever we procure a patient and a bit of tumor, and they grow up the cells for us, and then we're giving those back to patients. So those very personalized cellular therapies are coming, but as ever, that tends to be in the adult space first, and there's various reasons for that.
Yeah, just following on, is there any evidence that younger patients' T cells, for example, are just potentially going to be better than adults? Because we know that as you get older, your immune system and your T cells sort of slowly. So is there any evidence yet? Enough studies been done to say that the younger
no, because the numbers aren't big enough. I guess we're trying to do it at the other end of the spectrum. Do we get lower response rates in our very much older patients? Because as you say, there's that T cell exhaustion as you age, but they haven't seen the response rates. So I think the T cells are fit, but I'm not sure the tumors are getting the right signals across is the problem in the younger patients.
Yeah, interesting question, Andy. Thanks very much. Yeah, and that's another classic example where the trust are really, as Ruth would be the first to admit, through people like Tobias Minette doing some amazing stuff on the CAR T, which is being upscaled here, which is great. Thomas Butts, you had your hand up there, and I saw you nodding before in response to some of the comments being made. Did you want to come in there, Thomas?
Yeah, I did. My question was partially answered, I suppose, but I wondered. 2 really, really interesting stories, but an obvious distinction to me was the use with the medulloblastoma case of all of this wonderful genomic technology purely for stratifying quite old-fashioned therapies and quite devastating therapies. I wondered how far away you think in the round are we from personalized therapies or molecularly targeted therapies, not just diagnosed, across the board.
I mean, that's a great question, Tom, and probably has a number of underlying answers which all come together. So I think fundamentally children are different to adults, and we have the problem that many of the cancer-related pathways that are involved in childhood cancer development are key developmental pathways. So there have certainly been hits with agents that have shown good cancer regressions, but they've also shown quite significant developmental toxicities as well. So this is something we always have to keep in mind, the balance between the two. And of course, the younger the patient is, the more of a concern there is. I think we are, as you say, becoming good at stratifying conventional therapies, but they are still conventional. I think that will probably take the edge off the worst of the late effects, in that we can pick out groups that can have their treatment de-intensified. alas, many childhood cancers, they just don't have driver events that in the classic sense that we appreciate. And as a result, we don't really have the targeted therapies for them at the moment. So there's a huge amount going on to really try and understand the codependencies of what events we do know about. So for instance, let's say you've got a key translocation in leukemia. Can you understand just what the downstream pathways are that are targeted, such that they can be looked at with new therapies? But yeah, great question, and many intertwined answers, I'm sorry.
Great. Okay. Thanks, Tom. So I think we are coming to the end of the half hour. I'm very mindful of time because I really like people to keep coming back if they've enjoyed what they've heard this time. And these are really, really diverse talks, as those regulars will have ascertained. And obviously, if you do have any suggestions for other sessions, please do let us know. We'd love to hear from you. We're already in reactive mode for one of the forthcoming talks, actually, which we're going to feature on a very different topic, on the menopause. So there's a lightning talk going to be on that subject. So watch this space for that. So if you do have any other suggestions, please let us know, particularly where it looks like it's bridging across multiple partners, as the talk you've just heard clearly does, through both trust and university. So it just remains for me to thank you all for taking the time out of your diaries to attend, but particularly to Steve and to Ruth for putting together that really pacey and exciting talk to give a bit of a flavour of what's going on in cancer in Newcastle. And it's a lot, and it's a happening area. So thanks to you both. All right, take care, everyone. Thanks.